How do ecologists measure alpha diversity in biodiversity studies? Imagine walking into a lush forest and wondering just how many different kinds of plants and animals live there. That curiosity is at the heart of understanding biodiversity in a specific area. When ecologists want to find out how diverse a local community is, they look at what is called alpha diversity. This tells us about the variety of species in a particular place, like a pond, a grassland, or a forest patch. To do this, they use several tools and methods that help paint a clear picture of the local ecosystem. The simplest way to measure alpha diversity is by counting how many different species are present. This is called species richness. For example, if you find 10 different bird species in a park, that number gives you an idea of how many species are there. But just counting species doesn't tell us how individuals are spread out among those species. That's where species evenness comes in. It looks at whether one species dominates or if all species are equally common. Ecologists also use more detailed calculations called indices. The Shannon index is popular because it considers both the number of species and how evenly individuals are distributed. If the Shannon index is high, it means there are many species and they are fairly evenly spread. The Simpson's index is another tool that measures the chance of randomly picking two individuals from the community and finding they belong to the same species. A high Simpson's index indicates that a few species are very common, while others are rare. To get a better estimate of total species in an area, especially when some are hard to find, ecologists use the Chao estimator. It looks at the number of species found only once or twice and predicts how many species might be missing from the sample. Similarly, the abundance-based coverage estimator, or ACE, considers rare species to estimate the total number of species present. Another index called the Berger-Parker index measures how dominant the most common species is. If one species makes up most of the community, the Berger-Parker index will be high. Fisher's alpha is a bit different. It combines richness and evenness into a single number, especially when species follow a certain pattern of abundance. To make sure comparisons are fair, ecologists often standardize their data using a method called rarefaction. This adjusts 4 different sample sizes so that the diversity measures are comparable across sites. They also use visual tools like species accumulation curves, which show how many new species are found as sampling continues, or Venn diagrams that display overlaps between different samples. Measuring alpha diversity is a key part of understanding how healthy and complex an ecosystem is. It helps scientists see how things change over time, especially when environments are disturbed or restored. By counting species and analyzing their distribution, ecologists can better understand the health of local habitats and guide conservation efforts. In essence, these tools help break down the rich variety of life into understandable pieces, making it easier to protect and preserve biodiversity at the local level.